lipids are diverse compounds that consist mainly of carbon and hydrogen atoms linked by non-polar covalent bonds. It's any of various organic compounds that are insoluble in water, though some exceptions exist. They include fats, oils, wax, phospholipids, and steroids. Its function include storing energy, signaling, and acting as structural components of cell membranes. Lipids have application in the cosmetic and food industries as well as in nanotechnology.
Good day everyone, my name is Divino Francis C. De La Rosa, and I'm here to discuss the structure and classification of lipids. The structural formulas of these types of lipids illustrate the great structural diversity among the lipids. The defining parameter for lipids is a solubility rather than the structure. Here are the different examples of structure. We have a fat, biological waxes, steroids, glycerophospholipid, sphingophospholipid, and sphingoglycolipid. The common thread that ties all of the compounds together is solubility rather than the structure. All are insoluble in water. Two common methods exist for subclassifying lipids into families for the purpose of the study. Based on biochemical function, lipids are divided into 5 categories. We have energy storage lipids, membrane lipids, emulsification lipids, messenger lipids, and protective coating lipids. First is energy storage lipids. Triacylglycerols are stored in significant quantities as fat droplets in vertebrates, fat cells, and as oils in plant seeds. And the second one is membrane lipids, phospholipids, sphingoglycolipids, and cholesterol. Membrane lipids are a class of chemicals that make up the double layer surface of all cells, are structurally similar to fats. Cholesterol is an important lipid found in the cell membrane. It is a sterol, which means the cholesterol is a combination of steroid and alcohol. In the human body, cholesterol is synthesized in the liver. Emulsification lipids. Bile acids have a detergent effect, and dietary fat particles causing for globules to break down or emulsify into small droplets. Secretion bile and the role of bile acid in digestion. Bile contains bile acids which are critical for digestion and absorption of fats and fat-soluble vitamins in the small intestine. Many waste products, including bilirubin, are eliminated from the body by secretion into bile and elimination in feces. Messenger lipids, steroid, hormones, and eicosanoids. Our bodies possess chemical messenger known as hormones, which are basically organic compounds synthesized in gland and transported by a bloodstream to various tissues in order to trigger or hinder the desired process. How the steroid hormones act as messenger. The binding of a steroid hormone form a hormone receptors complex that affect gene expression in the nucleus of the target cell. Protective coating lipids. Biological waxes. Waxes are type of long-chain, non-polar lipid. Natural waxes are typical esters of fatty acids and long-chain alcohols. Waxes are synthesized by many animals and plants. Based upon whether or not saponification occurs when a lipid is placed in basic solution, lipids are divided into two categories. Saponifiable lipids. A saponifiable lipids comprise one or more esters groups, enabling it to undergo hydrolysis in the presence of a base acid or enzyme, including waxes, triglycerides, sphingolipids, and phospholipids. Non-saponifiable lipids. A non-saponifiable lipids cannot be disintegrated into smaller molecules through hydrolysis. Non-saponifiable lipids include cholesterol, prostaglandins. How are lipids important to our body? Lipids play a very important role in our body. They are the structural component of the cell's membrane. They help in providing energy and produce hormones in our body. They help in the proper digestion and absorption of food. They are a healthy part of our diet and taken in proper amount. They also play an important role in signaling.
Good day everyone! Today I'm going to discuss about types of fatty acids. There are two types of fatty acids, saturated and unsaturated fatty acids, which includes monounsaturated, polyunsaturated, and trans fatty acid. So in saturated fatty acids, all carbon atoms in the chain are saturated or bonded with hydrogen atoms, have no double bonds to other carbon atoms. This gives some molecules a little formation. Saturated fats are usually solid at room temperature, have high melting points. Foods are high saturated fats, includes pork, beef, cheese, whole milk, eggs, coconut oil, and butter. And unsaturated fatty acids have at least one double bonded set of carbon atoms. These double bonds can take two or more formation. It can be cis configuration or trans configuration. In the cis configuration, the hydrogen atoms on the same side on the double bonded carbon atoms, and the transfiguration, hydrogen atoms on opposite sides. Trans fatty acid, solid at room temperature, have high melting points. They're natural trans fats and artificial trans fats. Natural trans fats, also known as Roman trans fats, typically make up to 2% to 5% of dairy fat and 3% to 9% of beef fat. In artificial trans fats, turn liquid oils into solid fats to a process called hydronation. Hydronation is caused by vegetables, oils, or converted the solid fats simple by adding hydromatics. Some foods are contains trans fats include margarine, fried foods, and many fast foods. Monounsaturated fatty acids have a cis-molecular formation with the hydrogen same side of the double bond carbon atoms. This bent or kick formation, monounsaturated fats have one double bond in its structure. This usually liquid at room temperature have lower melting points. Polyunsaturated fatty acids also have a cis-molecular formation Again, the hydrogen atom are the same side of the double-bonded carbon atom. Also, given its kick formation, polyunsaturated fatty have more than one carbon double bond in its structure. They're typically liquid at room temperature, became solid when chilled, polysaturated acids. Generally classified by their omega numbering, the omega carbon is the carbon atom at the end of the hydrocarbon chain. There are 4 types of omega fatty acids: 3, 6, 7, 8, and 9. These determine by where location of the first double bond carbon atom is located. The fatty acid on the screen is the omega-3 fatty acids. Because the first double bond occurs the number 3, the others omega fatty follow the same instruction. And that is the types of fatty acids.
The physical properties of fatty acids, they are colorless, odorless, and tasteless. Solubility in water of fatty acids. Short-chain fatty acids are soluble. Long-chain fatty acids are insoluble. Melting point. It depends on length and chain of fatty acid. Short chain. It is unsaturated fatty acid with low melting point. Long chain, it is saturated fatty acid with high melting point. Insulation. Fat possess high insulating power. That means they are bad conductors of heat.
Organisms use lipids to store energy. Triacylglycerol are the main constituent of body fats and humans and other vertebrates, as well as vegetable fats, an ester derived from glycerol and 3 fatty acids. Their main role in cell is to store excess fatty acids, the transportation of fatty acids, as well as serving as an energy source.
Chemical reaction of triaglycerols, hydrolysis of triaglycerols. Energy production from triaglycerols starts with their hydrolysis into free fatty acids and glycerol. In adipose or fat storing tissue, this hydrolysis is carried out by a cellular lipase, which catalyze the hydrolysis reaction to release the free fatty acids and glycerol. The fatty acid is carried through the bloodstream by being absorbed to serum albumin, while the glycerol goes to the liver. In the liver, glycerols can be sent to the glycolytic pathway by the action of 2 enzymes, glycerol kinase and glycerol 3-phosphate hydrogenase. Glycerol 3-phosphate can be used as a source of glucose or after conversion to phosphonaphorate as a source of tricarboxylic acid cycle or TCA cycle intermediates. 3 common types of hydrolysis: salts is sodium plus water, 2 acetic acid plus sodium plus hydroxide, acid, acetic acid plus water, 2 acetate plus hydronium, base, ammonia plus water plus ammonium plus hydroxide. Saponification. Saponification is a process by which triglycerides are reacted with sodium or potassium hydroxide to produce glycerol and a fatty acid salt called salt. The glycerides are most often animal fats or vegetable oils. When sodium hydroxide is used, a hard soap is produced using potassium hydroxide results in a soft Based on the base used, soap can either be hard soap or soft soap. In general, potassium soap are not and sodium soap are hard. Visit cleaning action of soaps or detergent to know how dirt is removed from the objects. Saponification examples glycerols and soap. Soap molecule hydrophilic and attracted to water, hydrophobic and attracted to oil. Hydrogenation. Hydrogenation is a chemical reaction between molecular hydrogen or H2 and another compound or element, usually in the presence of a catalyst such as nickel, palladium, or platinum. Hydrogenation adds hydrogen across carbon molecule bands, increasing the degree of saturation. Partial hydrogenation is used to change the melting points of plants' oils. Some of the remaining double bonds are converted from CIS to trans, causing the products to have health effects similar to saturated fatty acids. Food labeled 0 grams trans fat per serving may contain oxidation. Oxidation is the loss of electrons of increase in oxidation state of an atom, ion, or molecule in a chemical reaction. Example, rusting of iron. And also, oxidation is essentially the oxidation of one of its constituents, fatty acids. It begins of isoprotides with the breakdown or lipolysis of triglycerides of the fatty acids and their glycerol. Often have objectionable odors, fats and oils are said to be rancid. Double bonds of triglycerols are subject to oxidation with oxygen in air, producing short-chain aldehydes or carboxylic acids. Antioxidants are added as preservatives.
Hi everyone, my name is Jada Conchino Romero. So for today's video, we will talk about membrane lipids. For this video, we will learn about what is membrane lipid, the function of membrane lipids, the properties of membrane lipids, the characteristic of membrane lipids, and the major classes of membrane lipids. So let's talk about what is membrane lipids. Membrane lipids are a group of compounds which form the double-layered surface of all cells. Membrane lipids are amphipathic, which is a molecule that possessed by hydrophobic and hydrophilic elements. When we say hydrophobic, substances that repels water, while hydrophilic having a strong affinity of water. The function of membrane lipids provide the physical environment where ion channel proteins go through complex conformational changes necessary for opening and closing. Membrane lipids are signaling molecules as chemical identifiers of specific membranes and as energy storage molecules. The properties of membrane lipids. Part of the lipid is ionic and therefore dissolves in water, where the rest has a hydrocarbon structure and therefore dissolves on the polar substances. Number 1. Cell membrane are thin enclosure that form close boundaries. Number 2. Cell membranes are made up of lipids, proteins, and carbohydrates. Number 3. Cell membrane consists of phospholipid bilayer. Number 4. Cell membrane are held together by non-covalent interaction. And lastly, the membrane are fluid-like structure. They are also characteristic of membrane lipids. Membrane lipids are principally two types, phospholipids and esterols. Generally, cholesterol Both types share the defining characteristic of lipids. They dissolve readily in organic solvents, but in addition, they both have a region that is attracted to and soluble in water. There are also three major classes of membrane lipids, which are phospholipids, glycolipids, and cholesterol. When we say phospholipid is a type of lipid molecule that is the main component of the cell membrane, while the cholesterol, a compound of the sterol type found in most body tissue. The last one, glycolipids, are lipids with a carbohydrates attached by a glycosidic covalent bond. Their role is to maintain the stability of the cell membrane and to facilitate cellular recognition.